Welcome to the deep dive. Our mission, as always, is pretty simple. Take complex research, cut through the jargon, and give you the knowledge that really matters.
And today we're looking at something quite fascinating. It's about peering into the past, specifically the viral past, to maybe predict future disease threats.
Right. We're talking about arboviruses. That
Yes.
stands for arthropod-borne viruses.
Exactly. These are the ones spread by, biting insects, mosquitoes, ticks, think dengue, Zika, West Nile, that group.
Nasty stuff. And they're a huge and actually a growing threat to global health, causing serious illness, even death, in both people and animals.
And there's a specific problem this research tackles head-on. A lot of the virus samples or isolates collected way back, say, the 1950s to the 80s, well, they lack full genomic data.
Because they were collected before the genomic era really kicked off.
Precisely. Sequencing a whole virus genome back then was incredibly hard and expensive. So researchers often got only bits and pieces, or they had to rely on older methods that weren't as precise.
But technology's changed. Since around, what, the early 2000s?
Yeah, roughly 2003 onwards, it became much faster, much cheaper. Suddenly, reading that entire viral blueprint was feasible.
So the mission here seems pretty logical. Take these old, potentially dangerous virus samples out of the freezer.
Right, 46 of them, collected across 23 different countries, spanning from 1954 to 1984.
And finally, sequence their full genomes.
Exactly. The idea was threefold. First, figure out exactly what their genomes look like. Second, update their classification, where they fit in the virus family tree.
And third,
And this is the crucial bit, use modern computer models to predict which of these old viruses might pose a risk of emerging, causing problems for us today.
okay, let's get into this then, because it sounds like more than just virology history. It's potentially about finding future threats, hiding in hiding in old test tubes anyway.
That's the core idea, yeah.
So before we dive into what they found in those old tubes, maybe let's just quickly underline the stakes. How big a deal are arboviruses right now?
Oh, they're a massive deal. And the problem's accelerating. Like we said, transmitted by mosquitoes, ticks, midges.
Blood feeders, basically.
Right, hematophagus, arthropods, technically, and the illnesses. They range hugely. Sometimes it's just a mild fever, hard to diagnose.
But sometimes it's much, much worse.
Absolutely. Severe stuff like neurological diseases, hemorrhagic fevers, think Ebola-like symptoms, though caused by different viruses, chronic arthritis that just won't quit, and even devastating birth defects like we saw with Zika.
And dengue.
Yeah.
You mentioned dengue earlier. The scale there is just mind-boggling.
It really is. Dengue is the heavyweight champ of human arboviruses. We're talking roughly 96 million cases every single year.
Wow.
And around 40,000 deaths annually. Plus, you've got nearly 4 billion people living in areas where they're at risk. It's more than half the planet.
An incredible public health challenge. And you said it's accelerating. What's driving that?
It's a mix of modern pressures, really. Things like unplanned, rapid urbanization, creating dense populations and breeding grounds.
More people, more travel.
Exactly. Increased human mobility spreads things faster. Then there's the uncontrolled expansion of the vectors themselves into new regions.
Mosquitoes and ticks moving around more.
Yeah, partly due to forest degradation, changing habitats, and the big one, climate change. Warmer temperatures, changing rainfall patterns. It lets these insects thrive in places they couldn't before.
Okay, so against that backdrop of this huge growing threat, getting better data, even historical data, seems critical.
Absolutely essential. Which brings us back to these 46 specific isolates. They came mainly from a big repository, the World Reference Center for Emerging Viruses and Arboviruses.
These are like libraries for viruses.
Kind of, yeah. Places that store and catalog these samples. And it's wild to think these particular ones were collected mostly in the 60s.
37% from the 60s, I read.
Right, and another 41% or so from the 70s, just sitting there in deep freeze for half a century.
Before we even knew sequencing would become, routine.
Exactly. And where did they get them from?
Mostly from the bugs themselves, right?
That's right. About 78%, nearly 4/5, were isolated directly from arthropods.
Mostly mosquitoes.
Yeah, mosquitoes were the top source, especially the Culex genus. They carry things like West Nile. But ticks were significant too, particularly Ixodes ticks, the ones famous for Lyme disease, though they carry many viruses too.
So we already know these viruses were circulating in vectors known to bite humans. The pathway was already there.
The potential pathway, yes.
Okay, so they sequenced these old viruses, and this is where it gets really interesting, you said, because the modern genomics basically rewrote some of the history books.
It really did. The old ways of classifying viruses, mainly using serology, well, the genomic data showed it sometimes got things fundamentally wrong.
Hold on, explain serology a bit more. Why was it prone to error? Is it like blood testing?
It involves blood serum, yeah. Serology basically looks at the immune reaction, how antibodies bind to the virus's surface proteins, its antigens. It's like testing if a key fits a lock based on shape and chemical interaction. But if the lock is a bit rusty, or if you have antibodies from a different but similar virus floating around that's called cross-reactivity, you might get a false positive. It looks like the key fits, but it's not quite right.
Ah, I see. So genomics.
Genomics doesn't guess based on the fit. It reads the entire blueprint of the key itself. Every notch, every ridge, it gives you, well, pretty much 100% certainty about what that virus is.
And when they read the blueprints for these 46, they found surprises.
Big time. First off, the diversity was impressive. These 46 isolates spanned 11 different genera across 8 viral families. That's a lot of variety.
And they found new viruses.
Based on the genomic data, yes. They proposed 15 entirely novel arbovirus species, essentially discovering new pathogens that had been collected decades ago.
Wow. Okay, so discovering new ones is huge. But what about correcting the old classifications? You mentioned discrepancies.
Right. There were 6 really key examples where the old serological classification just didn't match the genomic reality.
Give us one.
Okay, virus. Originally, it was classified in the Rhabdoviridae family, think rabies virus family, but the full genome sequence clearly placed it in the genus Orthobunyavirus, which belongs to a completely different family, Parabunyavidae. That's not just moving a branch on the tree, it's moving it to a different part of the forest entirely.
A major rewrite. You said there was another really illuminating one, Endel virus.
Ah, yes, Endel. This one's fascinating. It was originally typed as an arbovirus. Now, arboviruses are typically arboviruses spread by insects, so everyone assumed Endel was too.
And Genomic said.
Genomics placed it firmly within the mammalian orthorovirus group in the Samariviridae family.
Okay, and why is that such a big deal?
Because mammalian orthoroviruses aren't arboviruses. They typically spread through, well, think more like the common cold or stomach bugs, fecal-oral route, maybe respiratory droplets.
Wait. So for possibly 50 years, scientists might have been thinking this virus was spread by, say, a mosquito.
When actually it might be spreading through contaminated water or coughing.
Exactly. How does that happen?
Right.
How can you misclassify the entire mode of transmission for so long?
Because, going back to the analogy, the serological key just looked similar enough to other arbovirus keys to fool the initial test. There was enough surface resemblance.
But the underlying blueprint was totally different.
Totally different. Genomics corrected a fundamental assumption about its basic biology. It potentially stopped us from wasting time and resources searching for an insect vector that likely doesn't even exist for this virus.
Incredible. Okay, so the genome tells you what it is. Does it also tell you how it's built structurally?
Yes, in terms of organization. For example, the arboviruses they looked at had genomes broken into 8 to 10 linear double-stranded RNA segments.
Segments, like chapters in a book instead of one long scroll.
Good analogy. And the orthobunyaviruses, like that reassigned Kununura had the typical 3 segments L, M, and S of negative sense RNA. This segmentation is really important.
Why is that segmentation so important? You mentioned it leads to something called reassortment.
Exactly. Reassortment. It's a powerful evolutionary trick available to viruses with segmented genomes.
Okay, break that down. What is reassortment in practice?
Right. So imagine you have 2 different but related segmented viruses, let's say virus A and virus B, and they both infect the same cell in a host at the same time.
Like 2 different flu strains infecting one person.
Precisely like that. Inside that cell, as new virus particles are being assembled, they package up the RNA segments. But because both A and B segments are floating around, the new virus particles can accidentally grab a mix and match set.
So a new virus particle might end up with, say, the L and S segments from virus A, but the M segment from virus B.
Exactly. It's like shuffling genetic decks. This creates brand new combinations, potentially giving the virus new abilities overnight.
Abilities like adapting to a new host or a new type of mosquito.
Yes, or becoming more virulent, or evading the host's immune system more easily. It's a major shortcut for evolution compared to waiting for slow, random point mutations. It's a huge driver of how new viral threats emerge.
And did they see evidence of this reassortment happening in these historical samples?
They did. Clear examples. Ossa virus, one of the orthobunyaviruses, was identified as a reassortant.
How so?
It had its own unique M segment, but its S and L segments were clearly derived from a different virus called Madrid virus. They'd swapped parts.
And another one? Bruce Laguna virus.
Similar story. Bruce Laguna virus, another orthobuenyavirus, showed an M segment that clearly came from a different virus, the Alajuela virus.
So this swapping was definitely happening back then. It confirms these viruses have this potential for rapid, kind of unpredictable change.
Absolutely. It adds another layer of complexity and potential risk.
So this study also gave some context on specific viruses found, linking them to known diseases. You mentioned the arboviruses, Tinholmer and Bodine.
Yes, those clustered genetically with a species called Arbovirus magninsulae. Now, viruses in this group are typically tick-borne. They mainly infect seabirds, sometimes rodents, but critically related viruses in this group, specifically the Kemerovo complex, have been linked to serious neurological disease in humans.
So finding these relatives in the historical samples flags them as potentially concerning for human health?
It certainly warrants closer investigation, yes. They have relatives known to cause trouble.
And what about the bunya viruses? There was one called Xingu virus.
Right, Xingu virus. This one was proposed as a completely new orthobunyavirus species, and the context is worrying. It was originally isolated from fatal human cases in Brazil.
Fatal cases. What were the symptoms?
They presented with severe hepatitis, liver inflammation, but here's the kicker. Despite causing fatal disease in humans, we still have no idea what insect vector actually carries and transmits Xingu virus.
So a deadly virus, and we don't even know how people get it.
Exactly. It perfectly highlights the danger posed by these poorly understood, undercharacterized viruses potentially lurking out there. Okay, so we've now got these historical viruses properly classified. We know some are novel, some are reassortants, some have links to known human pathogens. What does this actually mean for, preventing the next pandemic?
Right, the prediction part. The study didn't just classify, they tried to assess the future risk using a machine learning model, right?
That's correct. A pretty sophisticated model based on the virus's genome composition.
What does that mean, genome composition? What's the model looking at?
It analyzes features within the genetic code itself, things like codon usage bias, basically, how the virus spells its genetic words, and whether that spelling is compatible with how human cells read genetic words.
Okay, so compatibility with human biology.
Yeah, and also sequence similarity. Does parts of the viral genome look suspiciously similar to bits of human RNA transcripts? This could hint at ways the virus might interact with or hijack our cellular machinery.
So the AI is looking for viruses whose genomes seem primed or ready to infect humans.
That's a good way to put it, yes, calculating the potential for that jump.
And what did it find? Did it flag anything specific?
It certainly did. The results gave us a kind of prioritized watch list. One virus stood out with the highest predictive score classified as having very high potential for human infection.
And that was?
Langen virus.
Langen virus. Tell me about that one.
Well, it's not new in the sense of just being discovered. It was originally isolated way back in 1960.
1960? Wow.
Yeah, from Dermacentor auratus ticks in Malaysia. Based on the new genomic data, it's proposed as a new species within the Viveros genus.
It's just amazing. A virus sample sitting in a freezer for over 60 years, and now an algorithm flags it as potentially the highest risk on this list.
It really highlights the value of going back and analyzing these historical collections with modern tools.
Okay, so Langen was very high potential. What about others?
Well, a significant chunk, 26 out of the 46 isolates, so over half, were classified as having a high potential for human infection.
Still very concerning. Any standouts in that high potential group?
The model gave the highest scores in that tier to Zingalamo virus, and then those two orboviruses we mentioned earlier, Tinholmer and Balin.
Zingalamo. That name rings a bell from earlier.
Yes, Zingalamo is genetically very close to Simliki forest virus, or SFV.
And SFV is known to be dangerous.
It is. It's been linked to fatal encephalitis in humans. There was actually a well-known case of a laboratory-acquired infection in Germany that resulted in death. So finding a close relative like Zingalamo rated as high potential is definitely cause for concern. It suggests it might have similar pathogenic capabilities.
And linking this risk back to the source, where did most of these high-risk viruses come from originally?
That's another key finding. Over 80%, 80.8% to be exact, of the viruses classified as high or very high risk were originally sampled directly from arthropod vectors.
Mosquitoes and ticks again.
Mosquitoes and ticks. So it's not like these are viruses looking for a ride. They already have an established connection to vectors that bite humans. The potential transmission route is already there.
Now, we have to be careful here, right? This model is based only on the genome. That's important.
Absolutely crucial caveat. The model predicts potential based on the virus's genetic makeup. But whether a virus actually emerges and causes an epidemic depends hugely on ecological factors.
Stuff the genome can't predict.
Exactly. Things like, is the vector present in large numbers near susceptible human populations? Are there changes in land use bringing people and vectors into closer contact? Unplanned urbanization, climate change, expanding vector ranges. These ecological opportunities are critical.
And they're inherently unpredictable.
Completely. Look at Zika. It was discovered in 1947. For decades, it caused only sporadic, mild illness. Nobody saw it as a major threat based on its history.
Until it suddenly wasn't mild anymore.
Right. Then, starting around 2007, driven by factors we still don't fully understand, maybe viral evolution, maybe environmental changes, likely a mix that exploded globally, causing microcephaly and other severe issues, Zika proves that ecological context can suddenly turn a seemingly low-risk virus into a global emergency, regardless of its long history. Genomics gives us potential, ecology provides the spark.
Okay, so pulling it all together, this research really did a lot. They went back to these historical freezers.
Characterized 46 old isolates, yeah.
Oh.
Using modern genomic tools.
Proposed 15 entirely new virus species based on that data.
And, importantly, corrected some significant classification errors from the past. The Undel virus story stands out, realizing it probably wasn't an arbovirus at all. That's a big shift in thinking.
Right. But maybe the most actionable outcome is that prioritized list.
I think so. We now have specific viruses, Langen virus flagged as very high potential, plus Zingalamo, Tinhomer, Bogaline as high potential that urgently need more research. Biological investigation, vector studies, checking if they're circulating now.
It gives surveillance programs concrete targets identified purely by reading their genetic code.
Exactly. It's using historical data to inform present-day preparedness.
Which brings us to our final thought for you, the listener. We just talked about Zika, how it was known but obscure for decades before suddenly catastrophically emerging. Now, this study flags Langen virus, isolated way back in 1960, as having very high predicted potential for human infection based solely on its genome. So here's the question. What responsibility do global health surveillance systems now have to actively look for Langen and these other newly identified high-risk historical viruses in current mosquito and tick populations, especially given that climate change is constantly shifting where these vectors can live? Should we be proactively hunting for these specific ghosts from the past before they potentially emerge?
That's definitely something to think about. How proactive should we be based on this kind of predictive genomic risk assessment?
Something for you